The anatomy of eukaryotic genomes. Two features of eukaryotic genomes present a major information processing challenge. First, the typical multicellular eukaryotic genome is much larger than that of a prokaryotic cell. Second, the cell specialization limits the expression of many genes to specific cells. The estimated genes, around 25,000 in the human genome, include an enormous amount of DNA that does not code for RNA or protein, but it is elaborately organized. Humans are fairly typical eukaryotes, and the human genome is a good model for eukaryotic genomes. All of the eukaryotic nuclear genomes is divided into two or more linear DNA molecules, each contained in a different chromosome. All eukaryotes also possess smaller circular mitochondrial genomes. The only general eukaryotic feature that is not illustrated by the human genome is the presence in plants and other photosynthetic organisms of a third genome located in the chloroplast. The basic physical structures of all eukaryotic nuclear genomes are similar, but the genome size is different in different organisms. For example, the smallest eukaryotic genome is less than 10 MB in length and the largest over 1 lakh MB. As can be seen in the table 1, this size range coincides to a certain extent with the complexity of the organism. The simplest eukaryotes, such as the fungi, have the smallest genomes, and the higher eukaryotes, such as the vertebrates and flowering plants, have the largest ones. The difference in genome size appears to make the complexity of an organism. Higher eukaryotes need larger genomes to accommodate the extra genes. It is also related to the number of genes in its genome. We have already learned that the human genome is split into two components, the nuclear genome and the mitochondrial genome, as depicted in figure 6. The typical pattern for most of the eukaryotes, the bulk of the genome being contained in the chromosomes, in the cell nucleus, and a much smaller part located in the mitochondria. And in the case of photosynthetic organisms, they are located in the chloroplasts. Let us now discuss the eukaryotic nuclear genomes. The nuclear genomes in eukaryotes are split into a set of linear DNA molecules, each contained in a chromosome. The studies in eukaryotes revealed that at least 2 chromosomes and the DNA molecules are always linear. The only variability at this level of eukaryotic genome structure lies with chromosome number, which is unrelated to the biological features of the eukaryotes. For example, Saccharomyces cerevisiae has 16 chromosomes, which is 4 times as many as Drosophila melanogaster. Sometimes the chromosome number is linked to the genome size. For example, salamanders, which have genomes 30 times bigger than the human version, but split into half the number of chromosomes. Coming to the packaging of DNA into chromosomes, chromosomes are much shorter than the DNA molecules that they contain. The average human chromosome has just fewer than 5 centimeter of DNA. Therefore, a highly organized packaging system is needed to fit a DNA molecule into its chromosome, because the nature of the packaging has an influence on the processes involved in the expression of individual genes. In the early 1970s, the important breakthroughs in understanding DNA packaging were made by a combination of biochemical analysis and electron microscopy. It was already known that nuclear DNA is associated with DNA binding proteins, which are called histones, but the exact nature of the association had not been delineated. In 1973-74, several groups carried out nuclease protection experiments on chromatin, which are DNA histone complexes that had been extracted from nuclei to retain as much of the chromatin structure as possible. In a nuclease protection experiment, the complex is treated with an enzyme which cuts the DNA at positions that are not protected by attachment to a protein. The sizes of the resulting DNA fragments indicate the protein complex's position on the original DNA molecule. After limited nuclease treatment of purified chromatin, the bulk of the DNA fragments have lengths of approximately 200 base pairs and multiples thereof, suggesting a regular spacing of histone proteins along the DNA. In 1974, these biochemical results were supplemented by electron micrographs of purified chromatin, which enabled the regular spacing inferred by the protection experiments that were to be visualized as beads of protein on a string of DNA, as shown in figure 6. Further, biochemical analysis indicated that each bead or nucleosome contains 8 histone protein molecules, 2 each of histones H2A, H2B, H3 and H4. Structural studies have shown that these 8 proteins form a barrel shaped core octamer with the DNA wound twice around the outside. The nucleosome particles are associated with 140 to 150 base pairs of DNA depending upon the species, and each nucleosome is separated by 50 to 70 base pairs of linker DNA, giving the repeat length of 190 to 220 base pairs. The beads on a string structure is the unpacked form of chromatin that occurs quite infrequently in the living nuclei. The cell breakage techniques developed in the mid-70s resulted in a more condensed version of the complex called the 30 nanometer fiber, the solenoid model for the 30 nanometer chromatin fiber, in which the beads on a string structure of chromatin is condensed by winding the nucleosomes into a helix with 6 nucleosomes per turn. The exact way in which nucleosomes associate to form the 30 nanometer fiber is not known. Several models have been proposed, the most popular of which is the solenoid structure. The individual nucleosomes within the 30 nanometer fiber may be held together by interactions between the linker histones, or the attachments may involve the core histones whose protein tails extend outside the nucleosome. The latter hypothesis is attractive because of the chemical modification of these tails result in the 30 nanometer fiber opening up, enabling the genes contained within to be activated. Elements of a eukaryotic nuclear genome: chromosomes, which are linear centromeres, telomeres, origins of replication and replicons; the protein, coding genes and spliceosomal introns; genes for non-coding RNAs, ribosomal RNAs, transfer RNAs, small nuclear RNAs, small nuclear RNAs and microRNAs; pseudogenes and process pseudogenes; mobile genetic elements and their remnants; satellite DNAs, micro mini satellites and repeated sequences; and fragments of organelle DNAs, nuclear mitochondrial DNA segments as well as nuclear plastid DNA segments. Coming to the eukaryotic organelle genomes, mitochondrial and chloroplast genomes, some genes might be located outside the nucleus. They are known as extrachromosomal genes and were first traced in the 1950s as a means of explaining the unusual inheritance patterns of certain genes that were found in the fungus Neurospora crassae, yeast Saccharomyces cerevisiae and the photosynthetic algae Chlamydomonas reinhardti. Electron microscopy and biochemical studies provided the evidence that DNA molecules might be present in mitochondria and chloroplasts. Eventually, in the early 1960s, these evidence were brought together and the existence of mitochondrial and chloroplast genomes independent of and distinct from the nuclear genome was accepted. Let us now have a look at the physical features of organelle genomes. Almost all eukaryotes have mitochondrial genomes and all photosynthetic eukaryotes have the chloroplast genomes. Initially, it was thought that all organelle genomes were circular DNA molecules. Electron microscopy studies revealed the presence of both circular and linear DNA, example in Paramecium, Chlamydomonas and several yeast genomes in some organelles. In many eukaryotes, the circular genomes coexist in the organelles with linear genomes, but in the case of chloroplast with smaller circles that contain subcomponents of the genome as a whole, this pattern reaches its extreme in the marine algae, dinoflagellates, whose chloroplast genomes are split into many small circles and each contain a single gene. Copy numbers for organelle genomes are also not particularly well understood. Each human mitochondrion consists about 10 identical molecules, which means that there are about 8,000 per cell, but in Saccharomyces cerevisiae the total number is probably smaller, even less than 6,500, even though there may be over 100 genomes per mitochondrion. Photosynthetic organisms such as the Chlamydomonas have approximately 1000 chloroplast genomes per cell, which is 1/5 of the numbers present in higher plant cells. Chloroplast genomes have less variable sizes and most have a structure similar to the chloroplast genomes of rice plant. The size of mitochondrial genome is also variable and is unrelated to the complexity of the organism. Most multicellular animals have small mitochondrial genomes with a compact genetic organization, the genes being close together with very little space between them. The human mitochondrial genome at 16,569 base pairs is typical of this type. Lower eukaryotes such as the Saccharomyces cerevisiae as well as some flowering plants have larger and less compact mitochondrial genomes with a number of the genes containing introns. Since genomes are very complex, minimal genomes for single cell organisms as well as minimal genomes for multicellular organisms. List some of the significant or representative genomes. Virus, example porcine circovirus type 1, which is the smallest virus, has a genome size of 1.8 kilobase pairs. Virus bacteriophage MS2, the first sequenced RNA genome, has a genome size of 3.5 kb. Virus phage 5X174, which was the first sequenced DNA genome, has a genome size of 5.4 kilobase pairs. Virus megavirus has a genome size of 1.3 MB. Virus Pandoravirus salinus, which is the largest known viral genome, has a genome size of 2.47 MB. Among the bacterium, Nasuia deltocephalinicola strain NAS-ALF, which is the smallest non-viral genome, has a genome size of 112 kb. Bacterium Haemophilus influenzae, which was the first genome of a living organism to be sequenced, has a genome size of 1.8 MB. Bacterium Escherichia coli has a genome size of 4.6 MB. Bacterium Cyanobacterium Chlorochlorococcus species, which is the smallest known cyanobacterium genome, has a genome size of 1.7 MB. Bacterium Nostoc punctiforme, 7432 open reading frames, has a genome size of 9 MB. The amoeboid Polychaeus dubium, largest known genome, has a genome size of 670 GB. Among the plants, Genlesia tuberosa, the smallest recording flowering plant genome, has a genome size of 61 MB. Arabidopsis thaliana, which was the first plant genome to be sequenced, has a genome size of 157 MB. Populus trichocarpa, which was the first tree genome to be sequenced, has a genome size of 480 MB. Paris japonica, the Japanese native pale petal, has the largest plant genome known and the genome size is 150 GB. Among the mosses, Psychomitrella patens, the first genome of a bryophyte that was sequenced, has a genome size of 480 MB. The fungus yeast, Saccharomyces cerevisiae, was the first eukaryotic genome to be sequenced and has a genome size of 12.5 MB. Among the nematodes, Pratylenchus coffeae, the smallest animal genome known, has a genome size of 20 MB, whereas Sinorhabditis elegans, the first multicellular animal genome sequenced, has a genome size of 100 MB. Coming to insects, the Drosophila melanogaster fruit fly has a size variation based on the strain ranging from 175 to 80 MB and the genome size was 175 MB. Among the mammals, Homo sapiens, estimated genome size 3.2 billion base pairs and the genome size is 3.3 GB. The approximate number of genes is 20,000. Among the fishes that have been sequenced, Tetraodon nigroviridis, the type of puffer fishes, the smallest vertebrate genome known, estimated to be 340 MB to 385 MB. Protopterus aethiopicus, the marbled lungfish and the largest vertebrate genome known, has a genome size of 130 GB.